Oh, finally I got in. Sorry for being late. Had trouble getting in today. Are there any questions before we begin? Can somebody confirm that you can hear me?
We can hear you.
Great. In chapter 8, let's go to the syllabus. Today is August 15th. Oops, close that down. And we are supposed to be working on chapter 8, finishing it and beginning chapter 3. Let me remind you, your infectious disease project is due this Saturday. Any questions about what we're going to be covering? Any questions about anything? All right, I have marked that we were to hear. We discussed chemical mutagens, specifically nitrous acid, and how it acts as a mutagen, changing, binding to adenine, and then changing it so the adenine doesn't react like adenine, it acts like a guanine. All right, when we're talking about the frequency of mutation, you should realize that spontaneous mutation rate happens about one base in 10 to the ninth replicated bases. So the mutation rate is 1 in 10 to the ninth bases. As 1,000 bases make an average gene, that means that there's one gene going to be mutated in a million genes. And that doesn't sound very high, 1 in 1 million. But if you look at the human population, which is something like 8 billion, and I don't know how many children are born in a year, but it's probably more than six. I mean, it's probably more than a million children born in each year. I don't know how many it is, but I'm sure it's more than a million. It's probably close to a billion. But my point is that when we look at that and look at the entire numbers in the human population, that there are children born at least every year that are mutants. And that's just by spontaneous mutation. Any question about any of that? A mutagen will increase the mutation rate something like 10 to 1,000 times greater than this, which means that the mutation rate will be 1 in 1,000 replicated bases to 1 in 100,000, or 10 to the fifth replicated bases. Any question about any of that? The most common mutagen that humans are exposed to is, of course, cigarette smoke. And it is a good mutagen, too. All right, let's talk a little bit about identifying mutants. You can do this experimentally. You can test for mutants using positive or direct selection, or two, negative and indirect selection. A positive direct selection detects a mutant cell because they grow or appear differently. Like if somebody were to see that walking down the street, you'd say, yep, there goes a mutant. Any questions about that? Negative or indirect selection detects a mutant cell because they do not grow when they should. Now, I'll talk a little bit more about that. How you get negative or indirect selection. Let me blow this up so you can see it better. And I actually did this working with bacteria cell. You can get isolated colonies on a plate and then stamp it with a velvet stamp. It's just a velvet piece of cloth. And then the cells will get on the velvet. So this one here There's, I think, that one there, that one there. I guess that's that one there, and then that one's that one there. And then you take this velvet stamp and you stamp other plates, transferring the cells from this plate to the velvet to this plate. And then you grow up these plates and you will get colonies in the same location as this plate. So that one there is this one here. That's not quite correct. That looks like these three here are these three here. The point being that that one there comes from that stamp there. And you'll notice that there's a colony here on this plate with a media containing the amino acid histidine, and it is absent in this plate in a media lacking the amino acid histidine. So these cells are mutant and they will not grow in the absence of the amino acid histidine. Any question about any of that? Anyways, that's how you do a negative selection, looking for cells that do not grow, where these cells grow in this media, but they do not grow in that media. Any questions? All right. So that's it for mutation. Now let's talk about genetic recombination. Recombination is the exchange of genes between different DNA molecules, or in humans, we're going to call it chromosomes. Crossing over is where two DNA molecules or two chromosomes actually come together and pair up, and then they exchange pieces. This normally happens in meiosis, where the mother's chromosome, let me see if I remember what this one is. The mother's chromosome, which is the blue one, and the father's chromosome, the white one, pair up in meiosis, and then they literally cross over, and then they break right here, where the mother's chromosome, which they've changed the color now, the lowercase e, becomes the, well, maybe, let me think about this. Maybe this is the mother's chromosome, and then that's the HI, the capital HI, right there. And it's got the part of the father's chromosome recombined with it. And this happens in humans as well as any other species all the time, if it's a sexually reproducing species, where the mother's chromosome pairs up with the father's chrome during meiosis, and then they cross over right here, and then the DNA molecules or the chromosomes break right there. And then there's the mother's chromosome here, and it's recombined with part of the father's chromosome. And there's the father's chromosome there, and it's recombined with part of the mother's chromosome. This happens all the time for homologous chromosomes in meiosis, and that's because the mother's chromosome number 1 is similar to the father's chromosome number 1. And even if they're different genes, most of the alleles will be the same. There just will be usually one allele different between the mother's allele and the father's allele. Like for eye color right here, there's only one nucleotide base different between the bluish eyes and the brownish eyes. Any question about any of that? Well, here's a question for you. What is different between crossing over and recombination?
Isn't crossing over where you're exchanging pieces of the DNA, whereas recombination is you're doing new combinations of the genes?
Yeah, recombination is a little more general. It's the exchange of genes between any DNA pieces or any DNA molecules or any chromosomes. Crossing over is literally where the chromosomes cross over and then exchange parts. And this almost always happens among homologous chromosomes, meaning one is the mother's chromosome, the other is the father's chromosome. So crossing over is just a little bit more of a narrow definition. Recombination is a little more general. Like you could get the X chromosome, part of it breaking off and then combining with the Y chromosome. That would be recombination. It may not be crossing over. And then we'll talk about, is it transposition? Where a gene can move from one chromosome to another. That is recombination event, but it is not a crossing over event. Okay? Any question about any of that? So crossing over is a little more strict in its definition. All crossing over is recombination. Recombination is a little more general, and it does include crossing over as well as any other recombination between two DNA molecules. Recombination could also be where I mentioned that the part of the X chromosome breaks off and joins the Y chromosome. There is no crossing over because the X chromosome gets none of the Y chromosome. So that would be recombination, but not crossing over. All right, let's talk about genetic transfer. We've already mentioned that vertical gene transfer occurs during reproduction between generations of cells, like a parent cell to the offspring daughter cells, or between two parents, if we're talking about humans, and one offspring. So that is vertical gene transfer. Horizontal gene transfer is the transfer of genes between cells of the same generation, where genes leave a donor cell and then they enter a recipient cell. Horizontal gene transfer can happen in 3 different ways in bacteria. I think it can only happen really in 2 different ways in human cells. There's transformation, conjugation, and transduction that can happen in bacteria. We'll talk about this happening in E. coli because I'm most familiar with it in E. coli, but it can happen in bacteria. Transformation is where DNA literally leaves the donor cell, and that usually happens when the donor cell dies. And then we have a naked piece of DNA out here, meaning it's not in the cell. Come on, not getting that to move. Oh, I'm pushing the wrong button, that's why. So that DNA comes out of a dead cell, and then it can be taken up by a recipient cell. And that DNA can recombine with the chromosomal DNA and then can be expressed. Usually this DNA that is entering the recipient cell has to recombine with the chromosome before the donor DNA can be expressed. Okay. And what's important here is, like, let's say the lowercase a gene codes for drug resistance. The original recipient cell is not resistant to the antibiotic. And then we get the donor A, bring in the lowercase a, and now the cell can be transformed and change its phenotype. And now it has the lowercase a gene, which is expressed. And now this cell is resistant to the antibiotic. Any question about any of that? And that's bacterial transformation. That does not normally happen in human cells because human cells do not normally take up DNA and they do not normally run into an environment where they see DNA as well. But like say your skin cells, let's say you're swimming in the pool and you happen to come across a DNA molecule, which I guess came out of somebody's dead cell, your skin cell is not going to take that up. Okay. So transformation does not normally happen in human cells. It can happen in a lab, and we'll talk a little bit about that, where you grow the human cells in a lab and then you add the DNA and then it can be taken up by the cell. We'll talk a little bit about that. All right, transformation, where the cell takes up a piece of DNA molecule, gobbles it, gets some indigestion, and then it changes the cell's phenotype as that genes is expressed. Any question about any of that? That's transformation. Okay, conjugation, also called sexual conjugation, is where a cell, which is called an F plus cell, shown here, has a sex pilus, shown there. That sex pilus literally goes out from this cell and then finds an F minus cell, skewers it, and then acts as a winch to bring the two cells closer together. The two cells do not have to come close together so that they're touching because that sex pilus is hollow. And then the F plus cell has a plasmid, which is called the F plasmid, which will replicate, and then one copy of the plasmid will come across the sex pilus and then go into the F minus cell. Any questions about that? And so literally, DNA from the F plus cell can move into the F minus cell. And it's usually just the F plus plasma that is transferred. Conjugation this is happening in a gram-negative cell. Conjugation can happen in a gram-positive cell. It does not involve a sex pilus. It involves a mating bridge where two gram-positive cells can come together and then they attach, and the cell wall disappears right there and right there. And then the cell membrane can literally fuse, where this cell membrane right there can fuse to that one, and then the cell membrane actually runs around this cell and then that cell, meaning these two cytoplasms are now connected. That's important because the chromosome from this cell can then move across to this cell, or not a chromosome, but more likely a plasmid. Sorry, I meant a plasmid. We'll talk about plasmids in a little bit if you're not familiar with what a plasmid is. A plasmid is a small molecule of DNA. And then vice versa, a plasmid from this cell can move through the mating bridge to that cell. So mating bridges are found in gram-positive cells, and they allow gram-positive bacteria to go through conjugation. This is called sexual conjugation because in both cases, DNA moves from one cell into another cell. In the case of gram-negatives, it always moves from the F plus cell to the F minus. In the case of the mating bridge, it may happen from this cell to that cell or that cell to that cell. Any questions about any of that? All right, so in sexual conjugation, like I said, the sex pilus literally skewers an F minus cell and then acts as a winch to bring them together. They do not need to come this close together. That the sex pilus is hollow, and then that F plasmid, called an F factor here, can replicate itself and then send one copy of the plasmid across the sex pilus and into the F minus cell. this F minus cell now has the F plasmid. The F plasmid codes for construction of the sex pilus. So this F minus cell has the F plus plasmid in it and then starts construction of the sex pilus. So this cell changes its phenotype and will become an F plus cell. And once it grows its sex pilus, it can go out and skewer an F minus cell and then send the F plus plasmid from this cell into the F minus cell. Any questions about sexual conjugation? If you wonder why it's called sexual conjugation, you just need to ask your significant other. Humans do not have sexual conjugation, but we are a sexually reproducing species. And literally, the sperm will join with the egg. And so we have a lot of genes coming together. In fact, the offspring is half the parents, half of each of the parents. Any question about any of that? All right. Let's talk a little bit about transduction. Transduction involves a virus. We haven't talked about viruses yet. I think it's our next lesson. But a virus, what it does, it will join with a bacteria cell and then pump its genome into the bacteria cell. What this genome does is it wants to replicate itself and call the shots in the cell that it infects. And an easy way to call the shots in the cell is to express a gene to degrade the cell's chromosome. And then the only DNA in the cell that's functional is the viral DNA. Any questions about any of that? We'll talk more about that when we're talking about viruses, but this is just an introduction to explain transduction. The virus then uses its DNA to make copies of the viral DNA and the viral protein, which will go ahead and make the virus. The virus then will insert the replicated viral DNA into the virus and then lyse the cell, releasing the new virions, which will then go out and infect other bacteria cells. However, let me blow this up so you can see the colors better. Every once in a while, a virus makes a mistake, and a virus isn't terribly worried about a mistake because a virus has lots of offspring or copies of itself. You can see that the purple red is the viral DNA, which was inserted into these viruses. But this virus has a purple or blue purple DNA, which was inserted, and it is part of the degraded host cell's chromosome. And now when this virus infects a bacteria cell, it will inject its DNA into the bacteria, but this is not viral DNA. This is DNA from this cell. And then that DNA can recombine with the chromosome of this cell, and then that DNA can be expressed. This is important because if this happens to be, let's say, that piece of DNA right there, encoding a gene for antibiotic resistance, this cell, which was sensitive to antibiotics, has now gained the cell for antibiotic resistance. Any question about any of that? Now, in bacteria, transduction happens. In human cells, transduction probably does happen all the time in our nose cells, where when we get a bacterial infection in our nose, every once in a while, the virus will infect you with somebody else's DNA. And then that could recombine with the DNA of your nose cell and then be expressed. However, who cares? This is just one cell in your nose. It is not a germline cell, so that this DNA will not be passed on to future generations. Any question about any of that? My point is transduction very rarely happens in the human population. You'd have to have a virus infecting a germ cell, either a sperm or an egg cell, and then putting a piece of DNA from somebody else's cell into that cell. And so that doesn't happen very often. Let's just word that it very seldom, almost never happens in the human population. Any question about any of that? But it does happen in bacteria. So, oops, let me go back a bit here. Let me stop that. There we go. So let's talk about, let me back up to all three of them. How often do transformation, conjugation, and transduction happen in bacteria? No, I'm talking about for gram negatives like E. coli, just because I happen to know the answer, but it's fairly similar for gram positive cells. Open a new cell here, new file, I mean. There we go. Large print so you can see this. So spontaneous generation, why isn't that printing? Spontaneous generation, I got it here. Not generation, mutation. Happens about once in every 10 to the 6 genes. Let me see if I can do that. Up. Okay, copy that just to get it going. Up. No, I gotta stop that. There we go. Transformation in E. coli, or most gram negatives, happens about I got the number, never mind. About 1 in 10 to the 5 cells. Just copy that whole thing. Having trouble here. Conjugation also happens about 1 in 10 to the 5th cells. And transduction also happens about 1 in 10 to the 5th cell, meaning all of these, oops, don't have that right. All of these, transformation, conjugation, and transduction, happen about 10 times more frequently than spontaneous mutation. Any question about any of that? At least in E. coli and other gram negatives. I guess I don't want to say that. All right, let's talk a little bit about plasmids. I've already mentioned that a plasmid is a small circular piece of DNA outside of the chromosome. That's what we mean by extrachromosomal DNA. It does contain genes. However, shown here, you can see that you can count the number of genes on probably one hand, maybe two hands. It's very few genes on a plasmid, whereas a chromosome has thousands, maybe even hundreds of thousands of genes on the chromosome. So a chromosome is a much bigger piece of DNA and has many more genes. A plasmid also replicates independently from the cell's chromosome. In fact, I showed you that earlier, right here, where this F plasmid replicated independently of the chromosome. Replicated right here and then sent one copy over there. The chromosome did not replicate. Any question about any of that? Plasmids are important because they are much more likely to be exchanged by transformation, conjugation, or transduction than chromosomal DNA. Any question about any of that? So a cell is much more likely to exchange plasmid DNA than it would chromosomal DNA. You should know that there are different categories of plasmids, but you do not need to know what those categories are or the names of them. Any question about any of that? They're just different types of plasmids, so you should know that. All right, let's talk a little bit about transpositions. A transposon is a gene that literally jumps from one position in the DNA to another position in the DNA. And that could, like when we're talking about human chromosomes, it could jump from one chromosome to another chromosome. This is called a transposon, a segment of DNA that can move from one region of DNA to another. Let me blow this up. A transposon always includes the transposase gene. This is an enzyme that literally cuts the transposon right here and right there, and then it can find another piece of DNA or another region of the DNA to cut and then insert itself in. And the transposase gene also seals the cut, sort of like ligase. So a transposon always includes the transposase gene, and then it always has inverted repeats on the ends of the transposon. The inverted repeats are thought to help the segment of DNA find another piece of DNA and then recombine with that other piece of DNA. These inverted repeats are actually a repeat. Let me see if I can figure this out. Here we go. A, up on here, A, C, T, T, and we look. I thought I had, oh, there it is, A, C, T, T. I thought I had it and then I lost it because I guess I was reading it the other way. Any questions about that? So that's the minimum a transposon can be. A transposase gene and then inverted repeats. We have complex transposons that have extra DNA in the transposon. Transposons, complex transposons can carry other genes, like this transposon called TN5 for transposon number 5, carries the kanamycin resistance gene. And usually the complex transposons have the extra DNA in the middle, and then they have a functional transposon on each end, meaning there's the transposase gene, there's the transposase gene, and there's the inverted repeat and the inverted repeat, and then the inverted repeat and the inverted repeat. Any question about any of that? Transposons are of importance, at least in populations, because they allow new gene combinations to occur, where the transposon can move a piece of DNA from one region to another region, and then that puts two genes together in combination, which weren't together earlier. A transposon can also move from the chromosome to a plasmid, or from a plasmid to the chromosome. And why this is important is that plasmids in bacteria are much more likely to be exchanged between bacteria than chromosome DNA. And so if you put the kanamycin resistance gene in this plasmid, this kanamycin resistance gene is much more likely to be spread to other cells, bacteria cells, by either transformation, conjugation, or transduction. Any question about any of that? So I guess I should say that crossing over and transposition do not make new genes, do not make new genetic information, but they make new combinations of genes that weren't existing previously. Like when in crossing over recombination, you put the mother's blue-eyed allele with the father's red hair allele. And now these two alleles are combined and they're a combination, which the offspring will have and potentially pass on. Any question about any of that? So crossing over and transposition do not make new genetic variation, but they make new combinations of variations. What they're doing is they're shuffling up the genes in the population. Any question about any of that? A new genetic variation, meaning if the population does not have this genetic variation, the only way it can arise is by mutation. An example of that was 10,000 years ago, humans only had the brownish-eyed allele. So humans only had brown eyes or dark eyes or hazel eyes before 10,000 years ago. And then in Northern Europe, we had a spontaneous mutation where the bluish-eyed allele formed. And that was new genetic variation for the human population. All new genetic variation has to arise by spontaneous mutation. But once that genetic variation is in the population, genetic variation or genetic differences can spread among the population by transformation, conjugation, or transduction. And in E. coli or bacteria, the genetic variation is much more likely to spread by transformation, conjugation, or transduction than it will arise by spontaneous mutation. In fact, it'll happen 10 times more frequently. Genetic variation is important in populations because differences in the genetic makeup drive evolution. You don't have evolution if there's no genetic differences in a population. But if there is differences, and if natural selection can work on those differences, you will get evolution. Any question about any of that? Anyways, that's genes and evolution, how genes can evolve. By mutation, spreading around by transformation, conjugation, transduction, making new combinations by crossing over and transposition, and that will drive evolution via natural selection. Any question about any of that? All right, that's it for chapter 8. Let's move on to our next chapter. Hopefully I didn't shut that down. I think it's chapter 13, but I want to make sure. Yeah, it's chapter 13. Let me shut down chapter 8 if I haven't done that. All right, chapter 13 covers viruses, viroids, and prions. The major goals and the rough outline of this chapter are outlined here. Know the terms. What is a capsid? What's an envelope? What is a viral spike? What is a bacteriophage? What is a lytic virus? What is a lysogenic virus? What is a prophage? What is a provirus? What is a latent virus? And what is a persistent viral infection? And then lastly, what's a prion and what's a viroid? Any questions about the terms? All right, 2, understand viral characteristics, come to know their structure, and the classification of viruses. 3, understand about viral multiplication. We'll talk about several viral life cycles, and the life cycle will be how the virus replicates. And then lastly, understand about prions and viroids. Any questions about this major goal and rough outline? If not, let's say that the only goal a virus has is to replicate. How do viruses replicate? They replicate by invading their host cell and hijacking that cell, forcing the host cell to make viral copies. The virus copies will then escape, and then the viral life cycle can happen again. I have a little video talking about viral replication. Let's see if I can get that to work. There it goes. Well, slow. There we go. No commercial this time.
Bacteria. Interactive piracy is about to take place. D4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral molecules plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus ejects its precious cargo of genetic material to the cell, commanding it to produce a new batch of T bacteriophages. In a final act, bacterium is forced to self-destruct, using hundreds of viruses to kill again.
Okay, any questions about that? All right, let's talk about some general characteristics of viruses. Viruses are inert when they are not within their host cell, meaning they display little or no metabolic activity when a virus is outside its host cell. Viruses require a living host to multiply. So you can say that viruses are obligate intracellular parasites. A parasite is something that lives on something else, and viruses require their host cell to live. So obligate, must. Any question about that? Viruses multiply within a living cell by using the host cell's enzymes, nucleic acid, amino acids, ATP, and etc. A virus has no ATP generating mechanism. When they need ATP, they simply use the cell's ATP, meaning the host cell they've infected. A virus contains DNA or RNA, but never does it contain both. So a viral genome can be either DNA or RNA, but it is never both. Viruses also always have a protein coat surrounding the nucleic acid. So this is showing you the nucleic acid of a virus in blue, and it's the centralmost molecule. It will be surrounded by a protein coat of the virus. Some viruses are enclosed in an outer lipid envelope, which is outside the protein coat. Not all viruses have a lipid envelope, only some. Any question about any of that? The host range of a virus is the range of cells that the virus can infect. Most viruses have a narrow host range where they can only infect a few cells of one or a few species. An example would be HIV, which can infect human T cells, and I think macrophage cells and brain cells and a few other cells of humans, and it can infect a few cells of one species of chimpanzee, and that is the only cells that HIV can infect. It cannot infect your skin cells, for example, or your bone cells, and it does not infect, I don't know, like a dog cell. Any question about any of that? The host range is determined by a specific host attachment site, like certain things on the cell wall, the flagella, cellular factors necessary for and cellular factors necessary for viral multiplication. The molecules of the cell that the virus binds to are called host cell receptors. Let me blow this up so you can see that better. So what the virus will bind to is called the host cell receptor. All it is a molecule that the virus binds to. Now, viruses themselves have molecules that bind to the host cell receptor, and these are called either ligands or adhesions, depending on who the author is. So an adhesion is a molecule on the virus that both binds to the host cell receptor. And a ligand is the same molecule on the virus. Most viruses only infect one or a few species, and usually only a few cells of that species. Bacterial viruses are called bacteriophages, and that can be pronounced bacteriophages. Both pronunciations are correct. Sometimes that's shortened to phages. I'll try not to use phages, but there's at least one movie I'll be showing it. It uses that term. I'll try and use bacteriophages to make clear I'm talking about a bacterial virus. Any question about any of that? All right, let's take a look at the relative size of viruses. Their size and diameter ranges from about 20 nanometers to about 440 nanometers. That's 0.02 micrometers to 0.44 micrometers. Now, 0.44 micrometers, or 440 nanometers, is up to and a little bit, just a little bit, past the size of the smallest prokaryote cell. The smallest prokaryote cell that is believed to be viable is down to about 400 nanometers. So viruses can get as big as the smallest prokaryote cell, but most viruses are something in between, and that means they're about 1/10 or less size of a prokaryote cell. And there we have different sizes of viruses here, showing you to E. coli, a little bit larger cell than your average bacteria. And then E. coli, or a bacteria, compared to a red blood cell, which is a small human cell, which is about 10 times larger than most bacteria. And I'm talking about the diameter of a virus. The largest virus by diameter that we're going to talk about is megavirus, which is about 440 nanometers in diameter. The longest virus is actually Ebola, which is a very long and skinny virus. So it is longer than megavirus, but in diameter, it's smaller than megavirus. Any question about that? The smallest virus we have is, let me see if I can blow that up because I can't read it, is about 24 nanometers, but nobody's probably ever heard of bacteriophage MS2. You have heard, though, of, poliovirus. That's the smallest virus you've probably heard of. And it's just a little bit larger than bacteriophage MS2. It's very small at 30 nanometers. Any question about any of that? Shrink that down now. Up, I didn't want to do that. When we're talking about the viral structure, we're going to separate viruses into the virion, and that's a single mature, complete infectious virus particle. So when we're talking about viruses, we're usually talking about virions. Viruses have a nucleic acid as their genome, and that genome can be either DNA or RNA. It is never both. If it is, well, it doesn't really matter. If it's the genome of a virus, it can be either double-stranded or single-stranded. Double-stranded DNA is normal. That's normally what our DNA is. Single-stranded DNA does happen in our cells whenever the DNA is being replicated or transcribed. The DNA will be single-stranded. In some viruses, their genome is single-stranded DNA. Some viral genomes is double-stranded RNA. This is unheard of in our cells. Our cells do not have double-stranded RNA. Any question about that? And then some viral genomes are single-stranded RNA. If the genome is single-stranded RNA, it's important to state whether it is the positive single-strand RNA or the negative single-strand RNA. The positive single-strand RNA can be read by the ribosome. It will encode AUG. The negative single-strand RNA is the complementary strand to the positive strand, so it does not code AUG, and the ribosome cannot read it. Any questions about positive single-strand RNA and negative single-strand RNA? Viruses can have their genome either as a linear molecule or as a circular molecule. And this should be familiar to you because human chromosomes are linear, and as well as all eukaryote chromosomes are linear. And then bacteria chromosomes are circular. Any question about that? Some viruses can have their genome in several segments. There's only one I'm going to talk about. Influenza virus has their genome in several segments. And this should be familiar to you because the human genome is in several segments. We call each of those segments a chromosome. Any question about any of that? All right. The term chromosome is not usually used for viral DNA because a chromosome has both DNA and protein in it, and the viral genome is only DNA or is mostly DNA. All right, talking a little bit further about the viral structure, all viruses are surrounded by a protein coat, and that name, it's called the capsid. This protein coat has been cut away so that you can look inside the virus. So that should be covered. Blow this up so you can see this better. The protein coat will be composed of one or more capsomeres. In this virus, each of these little circles is a capsomere. And as you can see, this protein coat is composed of only one capsomere. Any question about any of that? Now let's look at a virus that has more than one capsomere. Actually, both of these do, but it's not obvious. So let me come over here. Let's look at that virus. This virus here will have a capsomere for the head, at least two capsomeres for the sheath, a capsomere for the base plate, maybe for the pin as well, and then a capsomere for the tail fibers. So however many capsomeres that one is, there will be different proteins making the capsomere in different parts of the virus. Any question about any of that? If the virus has no envelope, it is called a naked virus. So this virus is a naked virus. If the virus has an envelope, it is called an enveloped or enveloped virus. That is a lipid bilayer. It also has proteins and carbohydrates in it, but it's mainly a lipid bilayer. And the envelope will be external to the protein coat. If I blow this up, you might be able to see it. Right there is the viral genome. Right there is the capsomere, sort of a hexagon. And then here is the envelope. If the virus has an envelope, it may have glycoprotein spikes shown by these reddish purple spikes right there. The spikes are antigens projecting from the envelope surface. An antigen is a region of a virus or a region of a molecule, which is the outermost regions of the agent, which the host recognizes and then mounts an immune response against. So an antigen is just something the host recognizes as foreign and then mounts an immune response against. And that will always be an external molecule. And in this case, the spikes are the most external molecule, and so they will be antigens to a host when this virus infects a host cell, or a host organism, I should say. The spikes are also the region of the virus that bind to the host cell receptor molecules. meaning the outermost molecule, are going to be the ligands or the adhesions of the virus. And in this virus, the ligands or the adhesions will be on the spikes. Any question about any of that? If you ever have anyone talking about the importance of the spikes on COVID-19, it's because the spikes are what allow the COVID-19 to bind to our cells and then infect the cell. So the attachment and ligands are the outermost regions of the virus. So in this virus here, where are the adhesions and the ligands of the virus? Come on, it's pretty obvious. All right, think about that. I'm not going to mention it. You guys have to know that. All right. Let me state that the antigens for this cell will be the protein coat because it's the outermost molecules of the virus. When we're talking about viral morphology, that is the general shape of the virus. Viruses are classified on the basis of their capsid structure, and capsids take three general morphologies. There will be a helical virus, shown here. Come on, mouse. On the leftmost right there. And I think everyone can see the helix there. If anyone sees the helix there, you can point it out to me. I don't see it, but I don't know. And then the virus can be a polyhedral, many-sided virus. This is actually an electron microscopic image of a polyhedral virus. There's the artist's rendition, and that side's removed so you can see inside the virus. The polyhedron sort of looks like a complex dice. And then the third general shape is called a complex virus. This virus can take on essentially any shape. This virus, in fact, takes on the shape like the Apollo lunar lander. If you remember the Apollo mission or you know anything about it, this is a complex virus. This is a complex virus. And that's a complex virus. Any question about any of that? This virus here is bacteriophage T4. We'll talk a little bit about that because this virus has been studied more than any other virus. Virologists know more about bacteriophage T4 than they do any other virus. Any question about that? T4 belongs to the T-even family, meaning T2, T4, T6. They all have this general shape shown here as the artist's rendition with a capsid head. Inside the head is where the genome is. There's the sheath, and then there's the tail fiber and the base plate. And this part here being the tail, and that being the head. As I mentioned, T4 is the virus that's been studied the most. I think the reason why T4 was chosen to be studied so heavily is simply because T4 is a virus that infects E. coli, and E. coli was a cell that most microbiology labs had. And so they decided to study T4 just because it was a virus that they could easily grow and then study. Any question about any of that? All right, let's talk about viral infection and the host immune response. When an animal gets infected with a virus, the virus will infect that animal cell, and then that infection will stimulate the host to produce antibodies to that virus. The host can make antibodies to anything that the host recognizes as foreign, and if the host can recognize the virus as foreign, it'll make antibodies to it. These antibodies will recognize and bind to the viral surface antigens, meaning the antibodies will recognize and bind to what we call the antigens. This is a problem with some viruses because the viral proteins can change or mutate frequently. Therefore, different viral strains can therefore infect the same individual more than once in the individual's lifetime. Can anyone give me an example of a virus that can infect a person more than once in that individual's lifespan?
The flu,
Yeah, the flu
COVID.
and COVID-19. Those are the two examples I was thinking of. Both of these viruses change their antigens, their viral proteins, fairly frequently, in which case new strains can arise, which can then infect the same individual. The flu, maybe we'll talk a little bit about it, is even more rapid in change than COVID-19. And we'll talk a little bit about why that might be the case. With COVID-19, it's because the RNA polymerase is not very accurate, and so it frequently makes a mutation. You don't need to know that. I'm just telling you off the top of my head. All right, how are we doing time-wise here? Oh, we got lots of time. So let's talk about viral taxonomy. That is the classification of viruses. Viruses are grouped into families based on the type of nucleic acid they have, the replication strategy, and the viral morphology. Viruses are also, well, I guess we'll talk a little bit more about families. Viral families always end in viridae. So like the herpes family of viruses, its name is herpes viridae. And then there will be a genus that will be underneath the family, and the genus name will always end in a virus. And so for the herpes virus, we would call it the herpes genus, and we call that the herpes virus. And then there will be a viral species underneath the genus, and that would be a group of viruses sharing the same genetic information in ecological niche, meaning they share the same host. And that will be the viral species. Viral species have common English names, and that's all we have, meaning the poliovirus only has the poliovirus as its common English name. There is no Latin name for viruses. And surprisingly, all of the early virologists were English speaking. And then when non-English speakers started studying viruses, they simply adopted the English name. So the poliovirus is used worldwide to name the poliovirus. Any question about that? You guys should be happy about that because you don't need to know any Latin names for the species. You just need to know the common English name. And then viruses can have subspecies. Subspecies are designated by a number. An example would be poliovirus type 1 and poliovirus type 2. The subspecies are very important for the virus because the subspecies can give different characteristics or different phenotypes or different diseases. The best example of different diseases is herpes simplex type 1, which is a subspecies giving the cold sores, and then herpes simplex type 2, which is the same species but a different subspecies, giving genital sores. Even clearer example of the difference between the subspecies is poliovirus type 1 and poliovirus type 2. Poliovirus type 1 gives the disease polio. Poliovirus type 2 is used as a vaccine to prevent individuals from getting the polio disease. So one gives the disease, polio, and the other we use as a vaccine to prevent getting the disease, polio. Any question about any of that? And if you want to know, the subspecies 2 of poliovirus is the Sabin vaccine, or the live poliovirus vaccine. Any question about any of that? All right, I've already given a little bit of an example of the viral taxonomy. The family name for herpes family is Herpes viridae. The genus would be simplex virus, and that would be herpes simplex virus for the genus. The common name, we do have a common name, human herpes virus. However, the common name has not been widely adopted even by the medical community. So I'm not going to cover it very much, and we will actually use what most doctors use, human simplex virus, as well as most common people use human simplex virus. And then that's the species name, human simplex virus. And then there's two subspecies, herpes simplex 1 and then herpes simplex 2. And there's actually a herpes virus being shown here. Now, many viruses do have a grouping above the family. Let me see if I can remember what that is. King Peter's son came over from Germany. All right, so that would be over. So many viruses have an order, and that would be above the family, but very few viruses have a grouping higher than the order. And the reason for that is the viral genome is fairly small, and it changes very rapidly to, what's the word I'm looking for? To better infect the host. So it changes to evolve to the host cell it infects. And it's not very long piece of DNA or RNA. And so we have a hard time trying to figure out the past history of viruses. So we do not group viruses much above a category of an order of the virus. Like there's no phyla of a virus. We do group viruses into the kingdom they infect. And obviously animal viruses will be viruses that infect animal cells. Bacteria viruses will infect bacteria cells. So we do group them by the kingdom they infect, but we have no other classifications until you get down to about the classification of an order. And it's just that we do not know the past history of viruses when we study their DNA. And it's because the genome changes so quickly and it's so small. Any question about any of that? All right, you can grow a bacteria virus called the bacteriophage in the lab. What you do is you just plate out the bacteria on an agar plate. Let me blow this up so I can show it better. And wherever the cell is whitish, that's from, bacteria growing in a lawn, meaning the bacteria growing everywhere. And then you plate on a sample containing the virus, and you spread that virus around the plate. And where the virus binds to a bacteria cell, it will then replicate, lyse that cell, and then go out and infect other cells, giving rise to a plaque. And wherever you see the darkening, that is a plaque formed by a virus causing that plaque. And with time, the plaque will grow. You can count most of the plaques here, and you can see this is actually 3 plaques that fuse together. But that plaque is larger than that plaque or this plaque. What probably happened here is that initially, this virus initially infected the bacteria cell. Let me close this down. Oh. Go back. And this plaque here, which is much smaller, the bacteria cell was not originally near the, actually, let's use paint. It'll be better. It was not originally near the, where the virus plated. So here we have, I'm just going to draw a little virus there. And then we have a bacteria cell over here. Let's make it a rod. What had to happen is this cell had to divide and then divide further. And again, sooner or later, I'll get there. And then finally, when the cell divided here, it's now touching the phage. And so the phage can infect this cell and then replicate, and it will lyse that cell and be released. And then the neighboring cells will be infected, and that will give rise to a plaque. And why this, I'm trying to explain why that phage is probably smaller than, might blow that up again. Why that phage is probably smaller right there than that phage there. Because this phage initially infected the cell, and that one, we needed the bacteria to grow so that the bacteria will eventually touch the phage and then become infected. Any question about that? All right, so viral multiplication. A viral genome only contains a few of the genes needed by the virion to reproduce. How the virus replicates is it hijacks its host cell and then forces the host cell to make copies of the virus. So what happens is the virus infects the host cell, takes over the host cell's metabolic machinery, uses that machinery, using the host cell's ribosomes, tRNA, amino acids, ATP, some of the host cell enzymes to replicate the virus. The basic mechanism of viral multiplication is similar in all viruses. Bacteriophages have two ways that viruses can replicate. Remember, a bacteriophage is simply a bacterial virus. Bacteriophages can replicate in the lytic life cycle, where the host cell is lysed and killed, and they can also replicate in the lysogenic life cycle, where the host cell remains alive as long as the virus remains in the lysogenic life cycle. Any questions about any of that? All right, let's take a step back before we talk about the two viral life cycles and say that a new bacteriophage, meaning a new bacteria viruses, needs what in order to replicate the virus? They need to replicate the viral genome, and they need to transcribe the viral genes to make viral proteins. The viral proteins then need to get together with the replicated viral genome to make the new progeny virions. This case is showing for a double-stranded DNA virus, but it doesn't matter what the genome is. You still have to go through these steps. I think I will show you the lytic life cycle movie. I don't think this plays, but let me look at it. Yeah, it does play.
First, the phage particles stick to the surface of the bacterial cell.
I can't show
Bind
it
to
though.
specific sites called phage receptors. Next, the DNA of the phage is injected into the cell, as if from a syringe. The ends join, forming a circular molecule. Then, the injected DNA replicates inside the host bacterial cell, and phage heads and tails are synthesized. The phage DNA is then packaged into the heads, and the filled heads attached to the tails. Finally, the cell dissolves or lyses, liberating about 100 newly formed phage particles after 45 to 60 minutes.
Sorry about that.
First,
Oops,
the
this, we'll get rid of that. There we go. This computer can't play that right. Let me see if I can find another movie which will show you a picture.
Is a parasite. Attacks the host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault, attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
Okay, any questions about that? We'll watch the rest of this movie when we talk about the lysogenic life cycle, which we will not discuss today, but we will next Tuesday. Let's see, where was I? So the lytic life cycle of a bacteriophage follows 5 stages. First, there's attachment, then there's penetration, then there's biosynthesis, there's maturation, and then lastly, there's release of the newly replicated viruses. So in attachment, the virus literally attaches to the host cell. Go here. And the viral capsid will attach, and actually the adhesions are in the ends of these tail fibers. Let me blow that up. So there's the adhesions right there on the virus, and the receptors would be right here and right here. The virus actually gets rid of the cell wall underneath the virus, and that's attachment. The virus then will contract, and you can see the virus is longer there than there, and that acts like a syringe. When the virus contracts, it puts pressure on the head, which will inject the viral genome into the cell, and that's penetration, shown there. Next is biosynthesis, where the virus will take over the machinery of the host cell, usually by degrading the host cell's chromosome, and then using whatever the virus needs to replicate, and it'll replicate its genome as well as the viral proteins. And They will then self-assemble in maturation, and the viral proteins usually will simply come together on their own and self-assemble. And then they have a little enzyme that the virus encodes that actually stuffs DNA into the virus, into the head. I think before the head is assembled into the virus. And then lastly, the virus genome will transcribe a gene, which is ligase, not ligase, what the heck is that lytic enzyme called? Lysosome. Lysozyme. There it is, lysozyme, which is an enzyme which digests the cell wall of the bacteria, and then water can move into the cell, causing the cell membrane to expand, and it'll eventually burst, releasing the newly replicated virions. And that's the 5th stage release. Any questions about any of that? That's the lytic life cycle. There's 2 important terms in the lytic life cycle. The burst time, that's the time from initial infection of the host cell by the virus to the time that the cell bursts. Burst time. It averages 20 to 40 minutes, depending on the species and depending on the temperature. And then the burst size is the number of newly released virions from the cell. And that too will depend usually on the species of the virus, something between usually 50 to 200 new virions will be released, depending on the species, from the infected cell. Any questions about the lytic life cycle? Bacteriophage T4 follows the lytic life cycle. The lysogenic life cycle, I guess we will talk about that, is where the virus attaches to the cell and then infects its genome into the cell. And once that happens, the cell has to decide whether, not the cell, the virus has to decide whether to remain in the lysogenic life cycle or enter the lytic life cycle. If the cell enters the lytic life cycle, not the cell, the virus enters the lytic life cycle, it then follows the life cycle I just discussed with the lytic life cycle. So a lysogenic bacteriophage can follow the lysogenic life cycle, but it can follow the lytic life cycle. A lytic life cycle virus, meaning a lytic virus, can only follow the lytic life cycle. But a lysogenic virus can follow both the lytic life cycle and the lysogenic life cycle. I mentioned the bacteriophage T4 only has the lytic life cycle. Bacterial virus lambda is a lysogenic virus, and it can either follow the lysogenic life cycle or the lytic life cycle. Any question about any of that? All right. So the cell, when it first becomes infected, I'll come back to that slide, it has to decide immediately whether to stay in the lysogenic life cycle or enter the lytic life cycle. If it enters the lytic life cycle, it just replicates the virus, bursts the cell, and then infects other cells. And then once again, because it's a lysogenic virus, it has to decide immediately whether to enter the lytic life cycle or enter the lysogenic life cycle. If the cell enters the lysogenic life cycle, the viral genome will integrate into the host cell's DNA. And that will begin the lysogenic life cycle. In the lysogenic life cycle, the lytic life cycle genes are latent or inactive. And how that happens is there's a lysogenic gene, which is a repressor protein, and that repressor protein shuts down the lytic life cycle genes. The bacterial host cell is called a lysogenic cell, and it will remain alive for as long as the virus stays in the lysogenic life cycle. And then the two will live in harmony, sort of symbiosis, together, with the lysogenic life cycle living inside the lysogenic host cell. Any questions about any of that? All right, so that shows that if the cell decides to enter the lysogenic life cycle, the viral genome will integrate into the host chromosome. We call that a prophage when the viral DNA is integrated in the host cell's DNA. Now, the prophage will only replicate when the host cell replicates its chromosome. So when this cell goes through cell division, come on, mouse, it will replicate its chromosome, and then it will split into one end of the chromosome. One chromosome will move to one side of the cell, the other to the other side of the cell. And that chromosome, both chromosomes, will have the prophage replicated. And then the cell will split into two. And this can go through many cell divisions where the host cell can remain in lysogeny with the lysogenic virus. Every once in a while, for reasons which are not well understood, and they've been trying to understand this now for over 100 years, the lysogenic life cycle will stop, and then the cell will enter the lytic life cycle. When that happens, the prophage will excise itself from the host cell's chromosome and then enter the lytic life cycle, replicating the viral genome and the viral proteins, making new virions, and then lysing the cell. And that's the lytic life cycle. Like I said, they don't fully understand why a lysogenic virus switches to the lytic life cycle, but they do know one thing that can cause that switch, and that is if the DNA of this cell is damaged, the virus will switch from the lysogenic life cycle to the lytic life cycle. And it's presumed that the lysogenic virus is afraid that this cell may die because it has damaged DNA. And then it will enter the lytic life cycle and then make sure it replicates. Any question about any of that? All right, let's watch the rest of that movie that I was showing. Did I shut it down? It looks like I shut it down. Sorry about that.
Sex. Attacks the host. Single host cell. Cycles happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates and the destructive lytic cycle begins. population of phage. Invade their hosts.
All right. There is one problem in that movie.
In a way, the virus
Right here,
is a
you go on.
parasite. It attacks the acid it incorporates in the
And that's showing T4 virus. T4 virus is not a lysogenic virus. It's a lytic virus. But that's the only error in this movie. Any question about any of that? All right. Oops, wrong thing. Where are we? Here we go. The last thing I want to talk about, lysogenic life cycle. I've already talked about the repressor protein keeping dormant the lytic genes. Here's the last thing I want to talk about, and that is two consequences happen in a cell when lysogeny occurs. So when the host cell becomes infected with a lysogenic virus, the host cell will now become immune to infection by that same lysogenic virus. So this cell cannot become infected by another lysogenic virus. Any question about that? The second thing that can happen is phage conversion, where the phenotype of this cell can change. And the reason being is there's the DNA of this cell. It's going to gain new DNA from this virus, shown here, and these can give this cell a new phenotype. It can change the phenotype of this cell, and that is called phage conversion, where the host cell may exhibit new properties from the genes from the lysogenic virus. The best example of phage conversion is Corynebacterium diphtherium cannot produce the Corynebacterium toxin unless the bacteria is infected with a lysogenic virus, meaning the Corynebacterium diphtherium toxin is present on the genes of the virus. And so only a lysogenic cell can produce the Corynebacteria diphtherium toxin. Any question about any of that? All right. I think I'm going to stop it there. Next time, we're going to talk about animal viruses. These are viruses that infect animal cells. Any question about any of this? If not, I'm going to stop the class a little bit early, and I'll see you at 7 o'clock for the lab, assuming we have a lab. Wait a minute. I'm not sure we have a lab, so let me check that. Where do I have that? There it is.
The last lab we did on Tuesday.
Yeah, there is no lab today. I will be there from 7 to 7:15 to answer any questions. After 7:15, or the last student logs out, I will log out. So if you have a question, I will be in the lab from 7 to 7:15. Okay? All right. Remember, your infectious disease paper is due this Saturday.
Have a good night.
All right, you too.